Now we are going to start a lecture series. Followed after each presentation, we are going to have a Q&A session. So as our first speaker, I'd like to invite Professor Russell Foster. Professor Foster is the head of the Nuffield Lab of Ophthalmology and a director of Sleep and Circadian Research Institute in the Oxford University. Professor Foster will talk about the light, sleep, and circadian rhythms, biology to application. Professor?
Well, good morning and thank you very much. It's a great honor and a delight to be here. I'd like to thank Mr. Lee for organizing this symposium. And I think it's really very important that the academic scientific community and the industrial communities interact, collaborate to hopefully improve health and well-being for all of society. So what I'd like to talk about this morning is light, sleep, and circadian rhythms, biology to application. It's going to be a broad overview and hopefully an introduction to my colleagues here, Manuel and Octavio, who will take up some of the themes that I'll start this morning. I thought we would kick off with the eye as the organ of vision, and it's certainly something that Manuel will also talk about. But the reason I want to talk about the eye as an organ of vision is because how it relates to the regulation of circadian rhythms and sleep. And so a big chunk will be on the biology of circadian rhythms and sleep, and a little bit about what happens when these systems fall apart, and how critically important it is to regulate these systems appropriately with exposure to the light-dark cycle. Then give some examples from our own work, but mainly from others, on human responses to light, alertness, shifting the clock, different wavelengths of light, and sort of try and summarize what we know, and as you'll see, it's not a great deal. And then finish by addressing, for me, some key questions and where we might like to go in the future collaboratively with academia and industry. So let's kick off with the eye as the organ of vision. And the first point I'd like to make is that, of course, the eye has to perform two fundamentally different sensory tasks. The familiar function, of course, and we've studied this for hundreds of years, is classical image-forming vision, the ability to detect an object against its background, either in black and white or in color. But of course, the eye performs a very different light detecting task, which is as a brightness detector for the regulation of a broad range of biology, including circadian rhythms, sleep, and levels of alertness, or wakefulness, as I think Octavio calls it. And let's kick off by considering the eye as the organ of vision. This is a very familiar picture, I know, to many of you, but just to orientate you, light, of course, passes through the front of the eye. It hits the retina, and if we look at the retina in a little bit more detail, we see that it's composed of these critical layers. The photoreceptors, the rods and cones, are the visual photoreceptors, the inner retina of horizontal, bipolar, and amacrine cells, and then these go off to the ganglion cells, which form the optic nerve. Now, if we look at one of these photoreceptors in a bit more detail, one of the things that's very obvious is that they have these membrane stacks, a concentration of membranes, and in those membranes is the light-sensitive molecule, the photopigment, consisting of two essential component parts, a protein, which essentially forms a cage, an opsin molecule, and embedded within the middle of this protein cage is a very specific form of vitamin A. And the first stage of light detection is the absorption of a photon of light by 11-cis retinal, and it then undergoes a conformation change to the all-trans state. This changes the disposition of the alpha helices, the protein, which then ultimately gives rise to phototransduction. Now, a photopigment such as this has a classic absorption spectrum, as illustrated here. And the shape of this absorption spectrum is highly conserved. It's very typical of an opsin vitamin A pigment. But how do you get different spectral sensitivities? How do you get different pigments? Well, what you need to do is change some of the amino acids in this pocket here. It changes the stability of this 11-cis. And what happens is that you can then generate longer pigments or shorter pigments, absorbing short wavelength, medium wavelength, or long wavelength light. But the biochemistry is largely conserved. In humans, we have 4 visual photoreceptors, the rods, and 3 sorts of cones, and they have very distinctive absorption spectra. The rod is centered at 500. Then we have a short wavelength cone at around about 420, 430. We then have a long wavelength cone based at 530 and a second cone at 560. And that's our visual repertoire. Now, for the rods, they just work, they work alone. And essentially what they're doing is absorbing reflected light. And so this light here is absorbed and this light is reflected. And so our rod-mediated vision is a world of black and white. And this contrasts very markedly with our ability to see color and the cone visual pigments. And so we can transform this black and white scene into a colored scene. And if anybody here can't see that there's an 8, then you either know or you should know that you have a color vision deficiency. But the key question is, well, how do we see this? How do we manage to detect different colors, wavelengths, are absorbed and reflected from this particular object here? So let's look at that in a little bit of detail. So the key question is, how can we see this as a sort of a turquoise color? And that's because our short wavelength cone will be stimulated by about 32%, our long wavelength 530 by about 80%, and our long wavelength 560 by about 40%. And essentially what you're doing is you're looking at the different signal strengths from these different receptors, and that is then encoded in the brain as color. So essentially it's differential excitation of different photoreceptor channels, and the brain then works that out as color. And just to reinforce the point, how do we see this as orange? Well, in this case, there's almost no stimulation by the short wavelength cone, a little bit of stimulation by the 530, 15%, but more stimulation by the 560 at 40%. Again, the differential stimulation of these receptors is encoding color. Okay, so that's basically a brief introduction to the visual system and color vision. Now I'd like to move to circadian rhythms and sleep and really address this second function of the eye as a brightness detector. Okay, not to state the obvious, but we sit on a planet that revolves once every 24 hours. And this revolution of the Earth on its axis provides immense ecological complexity for every living creature on the planet. We, of course, have the light-dark, day-night cycle, but in addition to big changes in light and dark, of course, there's big changes in temperature, our exposure to predation, our ability to acquire food. During the day, we're acquiring food, we're metabolizing it, and we're burning it up in terms of energy. But of course, whilst we're asleep at night, we have to depend upon energy reserves. So the biology of day-active metabolism and night-active metabolism is fundamentally different. Our social interactions, of course, change profoundly across the day. Our sensory systems change massively across the day. And indeed, our exposure to infection. So the immune system is also being challenged differentially across the 24-hour day. And in response to this profoundly different 24-hour oscillation of our environment, we have evolved an internal representation of a clock, which can predict and anticipate these changes in the environment and then adjust physiology in advance of the changed conditions. So we have in the base of the brain, in the hypothalamus, a master biological clock called the SCN, or the suprachiasmatic nuclei, and it comprises about 50,000 individual clock cells, which then work together to perform this master clock. So we have an internal representation of a day ticking away within the base of the brain. But it's no use having an internal clock unless it's adjusted to the external world. The classic mismatch between the internal day and the external world will be jet lag. We eventually get over jet lag as a result of exposure to the light-dark cycle, which is detected exclusively in humans and all mammals by the eye. So the eye aligns the internal master clock to the external world, and then the master clock then coordinates the rhythmic activity of our physiology. Now, what we've discovered recently, this field has discovered recently, is that you not only have clock cells within the SCN, but essentially every cell in the body is capable of generating a circadian oscillation. So essentially what's going on is that signals from the master clock then coordinate the rhythmic biology of literally billions of individual cellular oscillators which are scattered through the organ systems of the body. And of course, what these organ systems are doing are adapting, changing our biology. And here's some examples of just hormonal changes. Here's melatonin, cortisol, and growth hormone. And the key point is here's cortisol, which declines throughout the day. Here's sleep time. It's low during much of our sleep, but rises in anticipation of activity first thing in the morning, and then again shows this beautiful oscillation. Growth hormone is almost exclusively released at night, and growth hormone is used for cellular repair and the repair of tissue damage. And of course, melatonin is beautifully circadian regulated. Now, the key thing is that these rhythms persist under constant conditions. If you or I went to a deep, dark cave, constant light, constant temperature, these oscillations would continue, but they would drift through time relative to the external world. It's the light-dark cycle that aligns this internal biology appropriately to the outside world. And of course, it's that alignment by light that we're going to be talking about today. So the body clock or circadian system anticipates and then fine-tunes physiology and behavior to the predictable demands of the 24-hour light-dark cycle. Now, one of the most obvious changes in our 24-hour changes in our biology is the sleep-wake cycle. And approximately 36% of our entire lives will be spent asleep. So that's a huge amount of time, and we'll talk about some of the biology that's going on whilst we sleep. But the way that we sleep, this individual is going to bed at around about 11 o'clock and getting up at around about 7:30, but this changes between different individuals, and this is called an individual chronotype. This person is about neutral, going to bed a little bit before midnight and getting up after 6 o'clock. But there are some people who are very, very morning types, so they're going to bed way before midnight and they're getting up before 6 a.m. And there are other individuals who are evening types or late types, going to bed way after midnight and getting up way after 6 o'clock in the morning. And it's very important, and I think it's important for the lighting industry to appreciate that these different chronotypes, these different body clock types, an individual's morningness, eveningness, or whatever, depends upon, first of all, our genetics. Subtle changes in some of the genes that make up the biological clock can be different in morning and evening people. Also, age and development. So young people, teenagers tend to be late. Elderly people tend to be early, so it changes as we develop. But critically, and we'll come back to this during the talk, light exposure can have a profound effect upon whether you're a morning or an evening type. And part of the problem of our society, of increasingly being delayed, is that we're getting exposed to light at a particular time of day. And that's a theme I'll take up later on. Okay, I think it's also important to talk about the importance of sleep. Sleep is often dismissed as some downtime with very little importance at all. But what we see during sleep is that the brain is developing memories, our ability to lay down information and then retrieve it, but not only essentially retrieval of information, but also the processing of information. If you want to come up with innovative solutions to complex problems, a night of sleep has been shown overwhelmingly and in a number of labs all over the world to really enhance your ability to come up and problem solve. I think we've all anecdotally experienced this. You've woken up in the morning and you've solved a problem. And we now understand to some extent how the brain is actually achieving this. But also emotional processing. If you're tired or well rested, can very much influence your ability to remember things of a particular emotional content. So tired people forget the good stuff, but remember the negative stuff. And that's been shown in some really elegant experiments from Sickold's lab fairly recently. So a lot of really important stuff is going on in the brain associated with brain function. But in addition, the removal of waste products. So, for example, the clearance of beta amyloid from the brain, which has been associated with Alzheimer's and dementia, has now been shown to be cleared during sleep, or partially cleared during sleep. In addition, we have the regulation of growth and repair, the replacing of energy reserves, and the rebuilding of metabolic pathways. It's really important to appreciate that so much of daytime function is dependent upon a good night of sleep. This is not an indulgence, but essential part of our biology. Now, no great surprise, the sleep-wake cycle is generated from multiple brain structures and all the key neurotransmitter systems. So the key point I want to make is that sleep is not sort of a single bit of the brain, but it involves multiple areas of the brain. And I would argue that good sleep is a metric of good brain health. And again, we can talk about that later on. Okay, in addition to all of this circuitry and this interaction of all these neurotransmitters, the sleep-wake cycle, and of course the sleep cycle can be divided into non-REM and REM, and there are of course multiple wake states. But the key thing is that all of that circuitry is being regulated by some key drivers. In our own society, social timing is absolutely critical. I suspect, hands up, who was woken up by an alarm clock this morning? Yeah, and I suspect some of you aren't actually putting your hand up at the back there. Some gentleman is nodding vigorously. And of course, so much of our sleep-wake activities are driven by social demands, essentially forcing a sleep-wake cycle. But there are two key biological drivers which we need to understand. The first is perhaps the most intuitive part about sleep, which is called the homeostatic drive, and basically means the longer you've been awake, the greater the sleep pressure, the greater the need for sleep. And we'll talk about that process a little bit in a moment. But the homeostatic drive must be aligned with the clock. The clock, this master clock in the brain, the SCN, is time-stamping everything and is essentially saying, now is the appropriate time to be awake, now is the appropriate time to be asleep. And as we discussed, the master clock is then coordinating the rhythmic activity of multiple clocks throughout the body. But let's spend a little bit of time looking at this interaction because it's really important to understand it in the context of sleep-wake regulation. So, as I said, the homeostatic drive is an increase in sleepiness. So the longer we've been awake, the greater the build-up of sleep. And this has been associated with the build-up of adenosine, amongst other things within the brain. The reason coffee is effective is that caffeine in coffee blocks the receptors that detect adenosine, and that's why you feel more awake. But of course, whilst we sleep, the adenosine and sleepiness is dissipated. So sleep pressure rises and then falls. But towards the early evening, our sleep pressure is really high, but we don't fall asleep because of the action of the biological clock. So let's now combine this with the action of the biological clock, and what we see is that fairly shortly after we wake, the SCN is providing a drive for wakefulness. It's not very high because the sleep pressure is not very high. But as we go through the day, sleep pressure is increasing because of the homeostatic drive, but it's counteracted by this biological clock saying, no, this is not the appropriate time to be asleep, stay awake. So sort of this evening when you're taking your first glass of wine, well, 6, 7 o'clock, perhaps some of you 3 o'clock, the sleep pressure is really high, but you don't fall asleep because the clock is keeping you awake. However, as we then proceed into sleep, the wakefulness drive declines, and then during sleep, the sleep pressure, the wake drive is low, but then as we approach morning, the wake pressure begins to increase, and of course we wake at this point here when the sleepiness drive is really low and the clock is saying now is the appropriate time to wake up. And so what we see is sleep somewhere around here, and the sleep is most sleep is most likely over here because the sleep pressure is very high and the wakefulness drive is very low. If we wake up here, we can easily fall back to sleep because of the high sleep pressure and low wake drive, but of course, trying to get back to sleep here is a real problem. Sleep pressure has declined and the wakefulness drive is kicking in. Now, the key point is that the biological clock is being aligned to the external light-dark cycle, as we'll discuss in a moment. And for sleep to be effective, the sleep-wake cycle to be effective, these two systems, the sleepiness drive, the homeostatic drive, and the circadian drive, need to be appropriately aligned. Okay, So we've talked about the clock and the homeostatic drive, and now really the meat of today is the way that the eye interacts with the biological clock, but also be aware that the eye is not only regulating internal time, but it's also sending direct projections to other structures within the brain which are regulating levels of alertness. There's also an additional loop which involves the regulation of the pineal and pineal melatonin, which is essentially being regulated by the eye, and the clock reaches the pineal, and melatonin is released at night, and it's a biological marker of the dark. It is not a sleep hormone. It can modulate the effects of sleep, but it is not driving sleep. And there's a lot of, we can discuss this later, but there's a lot of confusion in the literature about melatonin. It's a modulator. It's a biological marker of the dark. The reason I've gone on about all of this stuff for some time is that what I'll have introduced to you is immense complexity. We have all of those key neurotransmitters, all of those different circuits. We have the demands of the alarm clock. We have the homeostatic drive. If you're drinking masses of coffee, you're masking that homeostatic drive. If the clock is not appropriately aligned to the light-dark cycle, then the sleep-wake alignment and things are completely disrupted. If you're getting lots of light, for example, before you go to bed, this is increasing alertness and therefore reducing or increasing rather the time it takes to sleep. So essentially, this complexity means that the sleep-wake cycle is immensely vulnerable to disruption. And the sorts of disruption that you see are related to one's overall health, one's cognitive health, one's emotional and mental health, and of course, all of these are interconnected. And so the key point I want to make is that light detection by the eye, regulating internal time, is right at the heart of essential parts of our biology. And so paying attention to this biology is critical for not only just the circadian system, but for our health, our cognitive health, and indeed our emotional and mental health. And let me give you some examples of what happens when you get sleep-wake disruption or sleep and circadian rhythm disruption. Well, short-term disruption of the sort that most of us experience on a routine basis leads to a loss of attention, high levels of microsleeps. There are 100,000 crashes on the American freeway every year, and that's been associated with falling asleep at the wheel. There's very important emotional responses. Tired people tend to be impulsive, they don't have empathy, and they have a negative focus. So many tired co-workers, for example, fail to pick up the social signals from colleagues and do stupid and impulsive things. You've also got memory impairment and increased mistakes, and critically, within the business sector, reduced cognition and creativity. This is endemic in much of the developed and the developing world, and we just disregard it. Longer-term problems, as associated with night shift work, lead to immune suppression, increased infection and cancer risk, probably due to the reduction in the immune system, increased cardiovascular disease, risk of diabetes 2, metabolic syndrome, other metabolic abnormalities, and increased stimulant and sedative use. So many people are driving the waking day with things like coffee and other stimulants, if you're really naughty, nicotine, but then that's being reversed at night with alcohol consumption and other sedatives like sleeping tablets. And the key thing is that those sedatives are just that. They're sedatives. They do not provide a biological mimic for sleep, and they're actually impeding some of the critical things going on within the brain, such as memory consolidation and the processing of information. So there are long-term risks. But also, if you are vulnerable to mental health states, whether that be mood instability, anxiety, paranoia, or hallucinations, or indeed have bipolar or schizophrenia, sleep disruption can nudge you further into the pathological state. And some papers we published just last year in Oxford, led by Dan Freeman, have shown that partial sleep stabilization in individuals with insomnia reduced levels of paranoia and delusional experiences. So there's a very intimate relationship between sleep disruption and mental health states. And we can talk about this in the discussion period. Okay, what I want to do now is strip back all of this and talk about this process here, the way that the eye is interacting with the master clock, and indeed the projections which are not shown, the way that the eye is also changing levels of alertness. So the key question we asked some time ago is, how does the eye detect the dawn-dusk cycle to regulate the circadian system? And this is a project I've been involved with for, certainly 25 years. It's a bit frightening. And just to reorientate you, what we're showing here is the light-dark cycle being detected by the retina, going to the SCN, and under these conditions, the light-dark cycle is regulating. This is a nocturnal animal, as it turns out, and so this individual is active, or this mouse is active during the night and asleep during the day. Doesn't really matter, it's just a cartoon. The key point is that if you have no master clock, and we've eliminated it here, then you are incapable of showing 24-hour patterns of activity and rest. But the key point for this story is that if you have no eyes, then you are simply unable to align internal time to the external world, and what happens is the clock then drifts through time, getting up earlier and earlier and earlier and earlier in this particular example. Okay, so the eyes are essential for circadian regulation. But what was clear some 25 years ago is that the way that the visual system and the circadian system processes or responds to light could not be more different. So, for example, in broad terms, the circadian system doesn't really care much where the light is coming from. It broadly captures light from a 180-degree arc, a field of view. But of course, for vision to work, it has to capture light in a particular region of space. You're building up a map of your light environment. Fundamentally different there. The circadian system is rather insensitive to dim light, whereas the visual system is ultra-sensitive to dim light. The circadian system really can only respond to long-duration stimuli, whereas, of course, the visual system is responding to ultra-short stimuli. And very critically, the circadian system has what's called long integration times. So it can count photons over long periods of time. In fact, over the minute to hour range, and we'll see some examples of that. Whereas, of course, the visual system, to function, it has to be grabbing light within the millisecond range. And what I couldn't understand as an early researcher was essentially, how can the retina perform both visual and circadian responses to light on the basis of our accepted and understood knowledge of how the eye was detecting light via the rods and the cones? So I will now condense about 20 years of work into a few slides. What we developed was a mouse, the rodless, coneless mouse, in which all of the photoreceptor cells had been genetically eliminated. And what was extraordinary is that these animals were completely visually blind. They had no visual responses at all. And yet, they could regulate their circadian system to the light-dark cycle perfectly normally with unattenuated light sensitivities. And this told us that there had to be something else within the eye, because if you covered the eyes or removed the eyes, this ability to detect the light-dark cycle had gone. So what the hell is it? If it's not the rods and cones, then we and others have been able to show that it's a new photoreceptor, photosensitive retinal ganglion cells. So those ganglion cells that form the optic nerve, about 1 in every 100 is directly light sensitive. And it's the receptor that is detecting the light information for the regulation of biological time. It uses a specialized photopigment, which is distributed throughout all compartments of the cell. And this absorption spectrum on opsin vitamin A has a peak at 480 nanometers in the blue part of the spectrum. And that's true for both mice and humans. So there's some confusion in the literature suggesting that the human peak is at 460. It's not, it's at 480. And of course, it's called melanopsin or OPN4. This is a gene that was originally discovered by Iggy Provencio from the melanophores of the African clawed toad, pigment cells in a frog. It's nothing to do with melatonin. It's from the origin of the word is melanophores, and this is an opsin extracted from those melanophores, and then was discovered in humans, and in fact, almost all other vertebrates. Okay. This is what one of those light-sensitive, photosensitive retinal ganglion cells look like, all these beautiful projections which are sensitive to light. And what you see at lower power is essentially lots of these ganglion cells which encompass the arc of the eye and act as this sort of photosensitive net, grabbing light and firing it off into the brain. Studies on humans were able to identify one individual who had lost all of her rods and cones, as far as we could tell, had a massively degenerate retina, and yet she was able to regulate her circadian system perfectly well. So humans are very much like mice. And in the same way, we have, yes, in lots of ways, actually. And also, the lambda max of human melanopsin is at 480 nanometers. And we can discuss why there's confusion in the literature. This blue, these PRGCs, are of the blue of a natural sky. Now, the key point is that the PRGCs regulate more than the circadian system. I've already alluded to this. And we, particularly Samahata, has been able to show that these ganglion cells project not just to the master clock, but multiple structures within the brain, which are associated with part of our pupil constriction, the livery pretectal nuclei, the master clock, as we've discussed, the sleep switch, the ventrolateral preoptic nuclei, the lateral hypothalamus, the subparaventricular zone. All key structures are associated with different sorts of biology. Furthermore, either via direct or indirect projections, they regulate the dopamine system, the serotonergic system, aspects of vision, and indeed mood as a result of projections to the medial amygdala. So this is a generalized irradiance detector system, not just a circadian photoreceptor system. And the final point is that the PRGCs don't always act alone. What's turned out to be really rather surprising is that the rods and cones can modulate the activity of these PRGCs in important ways. And so we know, for example, that the cones talk to the PRGCs directly via cone bipolar cells, but the rods also can communicate. There's a rod bipolar, which then communicates with an amacrine cell, which then talks to a cone bipolar, which can influence the activity of these PRGCs. This is really important because there's a complex and task-specific interaction between these different photoreceptor classes. And so we can't assume that blue light, because that's where these PRGCs are maximally sensitive, is just stimulating the PRGCs. And it's worth, I think, just illustrating that here. So here's the melanopsin peak at around about, it's got slightly switched, I'm afraid, 480. But it's also, yeah, I think something's got slightly distorted. But what we see here is a high stimulation of the rod, pretty good stimulation of the 430 cone, and less stimulation of the 530. But blue light is going to have a big effect on all the photoreceptor classes, as illustrated there. Okay, so let's now move to the third point of the talk, which is some human responses to light. And the first point to make, and this is something that Mr. Lee talked about, is that environmental lighting is very diverse. So we go from about 0.01 lux all the way to 100,000 lux. And to illustrate that, we have moonlight at this end, candlelight, sunset, and the museum display case is only about 50 lux. Office and modern lighting systems, about 300 to 600 lux if you're lucky. Near a window, you might get to 3,000 lux. Outdoor shade, 10,000 lux. And even in England, on a sunny day, you can get to about 100,000 lux. This admittedly is quite rare. Now, the photoreceptor classes that detect this light are the rods down here in night vision, but they saturate out at a fairly low intensity. Then the cones, which give us our day and full color vision, which can operate across the full dynamic range of natural light intensity. But the key point I want to make is that those PRGCs are relatively insensitive to light. They're in the sort of high hundreds to low 1,000 range. Now, I'm being vague about this because the key thing to appreciate is that the way in which these receptors respond to light will depend very much on the intensity of the light, of course, but its duration. And you'll see some examples of this later on. The wavelength of light. The time of day, dawn versus dusk, is profoundly different, and we'll see examples of that. What the sensory task is, it looks as though levels of alerting effects of light versus circadian light, the thresholds are different. The light history, if you've spent a lot of time outside versus inside, your sensitivities are going to be different. And as we've already touched upon, your age, whether you're young versus old, is going to have a big difference upon your sensitivity, the PRGC sensitivity. Now, our light exposure has changed profoundly over the past 200 years. This is an illustration for the United States showing that in 1800s, about almost 100% of the population was rural, and by 2000 it's approaching 20%. And in fact, it's probably lower than that in many industrialized countries. Which means, of course, that there's a progressive loss and attenuation of light as an entraining signal to the biological clock. So we're living in modern caves. As I said, at the window, you might be lucky enough to get 3,000 lux, but as you people know better than anybody, this rapidly attenuates as you move to the middle of the room, and it can drop from 3,000 lux to 50 lux over just a few meters. Modern settings, industrial settings, tend to be 300 to 600 lux. That's on the high end in many instances. And of course, even in our commutes, we're exposed to relatively low light in the underground systems. So there's a massive attenuation of light inside. Now, what we've got here is, of course, our domestic and commercial lighting systems are perfectly fine for the visual system, but they're actually, in many cases, below the threshold for the regulation of circadian responses to light. So the first point is, does natural versus artificial light alter human morningness versus eveningness? Does environmental light change our alignment with a light-dark cycle? And this is some work from Ken Wright's group. And what he did was to study students, and he maintained in their daily routines of work, school, and he measured their sleep onset and their sleep offset. And then he took them camping into the Rocky Mountains for a week. And he looked at the effect of this bright natural light on their circadian regulation. This is the week before, and this is the week after. And just to make this clear, he saw an advance of about 2 hours after natural light exposure. So yes, our hiding, and in fact it's worth pointing out, these individuals were getting actually quite a bit of light, almost 1,000 lux during the first 2 hours of wake, which is a lot more than many of us. And then this increased to about 3,000. And that increase in light intensity advanced the clock. So part of the reason for an increased late chronotype is the failure to see enough bright natural light. Let me expand upon that point. This is some studies that we now have that are in press. And this is a cartoon of the data. I won't sort of go through all the details. But the key point here is that the effects of dusk light versus dawn light are very different. If we see dusk at dusk, so if we see light at dusk, it'll make us get up later the next day. It delays the clock. But if we see light at dawn, it will advance the clock. We'll get up earlier the next day. So normally, if you're exposed to a natural light-dark cycle, you're sort of being delayed and then advanced. You're being nudged backwards and forwards so that you are appropriately aligned to the light-dark cycle. But of course, the big problem is that in many instances, we're not seeing the full light-dark cycle. Teenagers, for example, and this is the study that we'll publish soon, are essentially asleep. So they're missing dusk light. They're asleep, and so they're missing dawn light, so they're missing this getting up earlier signal. But of course, they're staying up late and getting lots of dusk light and missing out the dawn light, so they're only getting up the get up late signal, so they're being pushed to a later phase. So that's part of the problem that teenagers face. They're sleeping through dawn and they're being exposed to dusk. So getting the right light exposure at the home could make a major difference in the design of our lighting systems. This is another study. This is not by us, but from the Harvard group, led by Christian Kjochen, who's now in Basel. And this is the effects of light on alertness and sleepiness. And what he did was to keep people up and expose them to light from midnight to about 6:30 in the morning. And he compared two different light exposures. So what we're seeing here is alert on this part of the axis and tired here. And if the light exposure was about 3,000 lux, they stayed alert. But if it was low light, then you see a huge increase in tiredness. This is a good example of where light can have an acute effect upon alertness. This was expanded upon in this study, and we see increasing intensity along here and more alert along this axis. And what's important is that you see that you could get a 50% alerting response at 100 lux, and it saturates at 500 lux. So, people are saying, well, hang on, I thought you said the system is pretty insensitive to light. Well, it is relative to the visual system, and you only can get these responses if you give the light for 6.5 hours. So it's really important to appreciate that, yes, the system can be made fairly sensitive to light, but you need long-duration light exposure. And I think we, in the scientific community, have given you mixed messages. You're hearing that, oh, yeah, the system is actually rather sensitive. And then, of course, it's only sensitive if you get long-duration light exposure. It's something we'll come back to later. The effects of wavelength or color of light on alertness and sleepiness has also been looked at. And this is also by Christian Kjochen. And what Christian did was compare 3 different light sources, sort of a blue-enriched fluorescent lamp, an incandescent lamp, and a compact fluorescent lamp. So we go from blue light, in a sense, to orange light. Broadly the same photon flux. And what he showed was that sleepiness was reduced most effectively with the blue light and least effectively with the orange light. And also psychovigilance performance, the rate at which you can do particular tasks, was most effective under blue light and least effective under yellow and orange light. So the assumption has been, of course, that this blue light is being mediated via the PRGCs. But as I said, we cannot assume that the blue light will just stimulate those PRGCs. Remember what I showed you earlier. Those blue light wavelengths will stimulate the rod, the short wavelength and the long wavelength cones. So we don't know which receptors are actually mediating those effects of light on alertness. It's likely there's a major contribution from the PRGCs, but it's also highly likely, in my view, that the rods and cones are contributing. Should we be worried about reading ebooks before bed? Now, the press here has had a field day. And I think this is another example where I think we as the scientific community have sent a missed message. This is a very nice study. And what the individuals here, again from Harvard, did was compare an ebook on bright intensity, on its brightest intensity, versus the light reflected from a print book, as you see a much lower light intensity. And the methodology, and it's worth looking at the methodology, is that these individuals were looking at an ebook on full light intensity for 4 hours before bedtime on 5 consecutive nights. So this is a very serious stimulus. And what was the effect of that? Well, it delayed sleep onset by 10 minutes after 5 days. And it changed the amount of REM sleep by about 10 minutes over the same time. This was reported, however, as ebooks disrupt circadian rhythms before you go to bed. And this is really a gross misrepresentation of a very elegant study. And I think the problem is, this is a statistically significant effect, but it's a biologically irrelevant effect. I think we've, again, got to tease apart some of these issues. And finally, seasonal versus non-seasonal depression and the role of light. And this was a paper published last year which used some forms of light boxes. And this was on seasonal depression, but similar reports have been shown for seasonal affective disorder and SAD. This is a very nice paper published in 2016. And let me just go through the data. So these are the weeks of treatment. And the green here shows the placebo. This yellowy orange is the impact of fluoxetine, an antidepressant. This red line shows the effects of lightbox treatment, which was 30 minutes a day in the early morning of 100,000 lux. And what's very interesting is that this final line here is the lightbox treatment, so the light treatment, plus the fluoxetine. And what you see is the fluoxetine and the light were additive. So the light and the fluoxetine are probably acting via different mechanisms. But this provides really clear data that on its own, light is, for non-seasonal depression, is a critical way, the most effective way we have of getting rid of depression, or alleviating some of the symptoms of depression more accurately. Mechanisms, again, you'll hear masses about this in the literature, but despite much conjecture, the mechanisms are very poorly defined. Some people are saying it's all due to light and serotonin. We have no empirical evidence for that yet. Finally, and I really promised to have finished in just a few minutes, what are the key questions for me and the future research? Where might we work together? I think we really must take into consideration both visual light and circadian light levels in building design. But how do we do it? I think, again, it's a complicated issue. It's going to be PRGCs versus rods versus cones, and all of these factors, as we've discussed, will interact. And in my view, we don't have enough data really to model this. I think part of the response has been to say, well, we can model the effects and the contribution of these different photoreceptors, and that will tell us what lighting we need. I don't think we can do that yet. I think we need to do the following sets of experiments. We need to measure the direct impact of how much light, the type of light, I guess the wavelength, and when light exposure occurs, and we can measure 3 parameters. One is sleep-wake timing. The other will be alertness and cognition. Of course, we've got to be careful about these terms, as you'll hear later from Octavio. We're not consistent about how we use these terms. And then issues of overall well-being. So, we've shown this image before. Dusk light will delay the clock. Dawn light will advance the clock. So light exposure around dawn and dusk is really critical. Our activities vary immensely across the day, whether we're getting exposed to natural light on our commute to work, artificial light whilst we're at work. We may get out and see some more light. We may be stuck next to a window. We may be sitting under LED lights or fluorescent lights or incandescent lights. There may be light streaming in through the curtains when we're trying to sleep. And in the morning, we're standing in the most brightly lit room, which is the bathroom, getting quite a major pulse of light artificial light before we go outside. The key thing is, though, we've got to measure the impact of these different activities and these different light exposures. Where should the lighting industry concentrate its efforts to achieve optimum health and performance, home versus work or combined? And we simply don't know the answer to that. We can measure sleep-wake timing fairly easily. So you can strap one of these little things onto your wrist, and you can measure when people went to bed and when they got up. You can collect urine over 48 hours, and so you can measure sleep onset and sleep and wake. You can collect urine over 48 hours and measure the peak in melatonin to get an independent measure of where the biological clock is. You can also, with various devices, perhaps not that easily, but you can measure light levels, and in some cases some idea of the light spectrum of exposure across the day. And critically, one can measure different levels of brain activity, beta waves, awake, normal alert versus relaxed, versus deep relaxation and early sleep versus deep sleep. So EEG measures can provide measures of alertness and sleepiness. Now, up until fairly recently, the only easy way to do that was to put somebody in a lab and strap them down and put the electrodes all over their head. But in Oxford, Martin de Vos has devised a new way to measure EEG with a little device that fits behind the ear. It talks to a smartphone. And so we can go into the field, look at levels of alertness under normal levels of illumination, walking through the day, or look at the impact of disrupted sleep at night, again, using this little device. So we can go into the real world and measure levels of alertness and sleep. And then finally, of course, there are a large number of validated questionnaires which can measure mood and well-being. This happens to be one of the HAD scores, which is the Hospital Anxiety and Depression scores, and people can fill out these either in paper form or on a smartphone, and we can measure alertness and well-being using validated questionnaires. So my final slide is a plea, really, which is that I don't think we can develop human-centric lighting until we really know what impact light has upon human biology across the day-night cycle. I don't think we've got the data yet. And the final comment will be, we must define human lighting ecology in the urban environment. And I think once we have some empirical measures, we can then use evidence-based approaches to design lighting systems appropriately. Okay, so what I hope to give you in the last hour was a sense of the eye as the organ of vision, and contrast that with its role in regulating circadian rhythms and sleep, and really highlight why light is so important in stabilizing the sleep-wake cycle. Because if the sleep-wake cycle is disrupted, as we see in many of us and indeed in many shift workers, then health and well-being falls apart very rapidly. And then finishing by asking some sort of key questions, and really, what is light doing to human biology across the 24-hour day? And as soon as we know that, I think we can then use this information for better lighting design. And with that, I'll finish. Thank you for your attention.
We're a little behind the schedule. We're going to take 2 questions from the floor.
Gentleman there. That's a really good question. So napping, the data seems to be that if you take a 20-minute nap, you can, around about lunchtime, early afternoon, you can improve your performance during the second half of the day. But there's a danger with napping. If you go longer than 20 minutes, you can go into deep sleep, and the recovery from deeper sleep can leave you sort of groggy and counteract the beneficial effects of a nap. The problem with napping is, of course, what it will do is push back the sleep pressure, which means that potentially you'll go to bed later, which means that if the alarm clock is driving you out of bed, you've got restricted sleep. You've gone to bed later, but you're getting up at the same time, which means you wake up tired, which means that you need a nap, and you can get into a cycle of later and later bed and longer naps. And this is the classic problem we see in teenagers. So, for example, they are chronically sleep deprived. They're going to bed really late. They're struggling through school. They get home from school. They then take a 2-hour sleep. Can't be recalled a nap in many cases. That's massively pushed back the time at which they'll go to bed that night. And so you can really disrupt the sleep-wake cycle. And it's a good example where the homeostatic drive and the circadian drive are completely misaligned, and you get disrupted sleep. So the occasional 20-minute nap, fine. If you become dependent upon it, you're clearly not getting enough sleep at night, and there's something you need to do about it. Gentleman there. Yeah. So, of course, you're struggling with chronic tiredness, and so high sleep pressure, and the clock is completely the wrong phase. And it's worth bearing in mind, it's a good illustration that you're not simply 6 hours shifted from Frankfurt to New York. The whole circadian and sleep systems are in a complete biological smear of time. The brain wants to do something at one time, the gut another time, the muscles at another time, so you have this huge disruption. The key way to get over jet lag is so the rule of thumb is if you're traveling west is to seek out light. If you're traveling east more than 5 to 6 time zones, then you avoid morning light, but then seek out afternoon light. And that'll advance the clock to the new time zone much more effectively. Now, another talk I could have given will be the molecular dissection of these pathways. We actually know what causes jet lag at a molecular level. There's a protein in the clock. So light hits the clock, the molecular clock begins to move. And then another protein kicks in, which stops it moving effectively. So essentially, there's a break on the effects of light on the clock. So you can't move to a new time zone, which is 6 hours away, because there's actually mechanisms which prevent you doing that. There's a big buffer. When we've taken this protein out of a mouse and we've changed the light-dark cycle, the mouse completely realigns to the new light-dark cycle within a day. It's quite extraordinary. So there's some interesting biological mechanisms that we're working on and thinking about how we might manipulate for things like jet lag. Yes, that's it. Okay, thank you again for your attention.
Thank you for the great lecture, Professor Foster.